Welcome to the peaceful space of sleep-friendly science, where tonight we journey deep within the hidden architecture of our own cells to explore a mystery written in the very ink of life. Here, in this quiet sanctuary, we trade the noise of the day for the gentle hum of curiosity. Allow the world outside to soften and fade, like starlight filtering through a forest canopy. There is nothing to do, nothing to solve, simply a space to be, to listen, and to wonder. Now settle in, take a deep breath, and let the soft sounds guide you as we begin tonight's journey into the unknown. Section 1. The Illuminated Manuscript. Imagine, if you will, that you are not a body of flesh and blood, but a library. A living, breathing, ancient library of almost impossible scale. Within you, not one library but trillions of them, each one a single perfect cell, and each one holding a complete copy of the master text, the story of you. Float down past the shelves and corridors of your own being, and let us choose one of these libraries, one single cell. We drift through its outer wall, a soft and yielding membrane, into a bustling yet silent inner world. And in the very center of this world we find the most sacred room, the heart of the library itself. This is the nucleus, a quiet, domed chamber, and it is here that the books are kept. For a long time we, as scientists and storytellers, believed we understood the nature of this library. We saw the books, coiled and spooled into beautiful structures called chromosomes. We learned the language in which they were written, a simple, elegant alphabet of just 4 letters. T, C, and G. The ink of life. And we called the books themselves genes. This was a profoundly beautiful, satisfying idea. The library was orderly. Its purpose was clear. Each book, each gene was seen as a single, exquisitely written instruction. A recipe. A blueprint. Think of it like an illuminated manuscript from a time long past, painstakingly copied by scribes, with a clear and divine purpose. Each manuscript contained the complete instructions for building one specific vital thing. What did these books describe? They described the creation of proteins. Proteins are the true laborers of your cellular world. They are the microscopic machinery that makes you, you. If a gene is the blueprint, a protein is the cathedral built from it. A protein is the mason that lays the bricks of your bones, the weaver that creates the soft collagen in your skin, the messenger that carries whispers of thought from one nerve to another. They are the engines, the couriers, the architects, and the artists of your body. And so, the central story of life seems simple and direct. One gene, one book of instructions created one protein, one masterpiece of molecular engineering. Life was a grand collection of these exquisitely clear manuscripts. To understand life, we believed we simply needed to read all the books. This grand ambition led to one of the greatest scientific endeavors in human history, the Human Genome Project. It was a quest to be the ultimate librarian, to open every book on every shelf within the human nucleus and transcribe its every letter. For over a decade, scientists around the world worked to read this fundamental text, the complete genetic story of a human being. The anticipation was immense. How many books would we find in this library of humanity? Given our complexity, our thoughts, our dreams, our art, our consciousness, the number must surely be vast. The estimates were high, perhaps 100,000 genes, maybe more. 100,000 beautiful, clear instructions to account for the wonder of a human being. Finally, the work was done. The librarians had finished their catalog. And as the data was compiled, a quiet, collective shock rippled through the scientific world. A wave of profound humility. The number of books, the number of protein coding genes wasn't 100,000. It wasn't even close. It was only around 20,000. 20,000 blueprints. A rice plant has more. A simple water flea has more. The magnificent library of humanity, the source code for symphonies and skyscrapers, for poetry and philosophy, seemed to have fewer books than many far simpler organisms. This was the first great mystery. A whisper that our understanding of the library was fundamentally incomplete. But the true shock, the discovery that would set us on the path of tonight's journey, was not in what the librarians found, but in what they didn't find. As they had read through the long spools of DNA, they found that the beautiful, clear manuscripts, the genes were few and far between. They were like tiny scattered islands of meaning in a vast, seemingly empty ocean. Imagine opening a grand ancient tome, expecting every page to be filled with elegant script, only to find a single beautiful sentence on page 1, another on page 50, and another on page 200. And all the pages in between? They appeared to be filled with nonsense, gibberish, repetitive strings of letters with no discernible meaning, no clear instruction. This was the state of the human genome. The genes, the blueprints for proteins, made up a shockingly small fraction of our DNA, less than 2%. The other 98% was a mystery. It was a vast, dark, unknown territory. Scientists, in a moment of honest puzzlement, gave this enormous silent majority of our genetic code a simple, dismissive name. They called it junk DNA. For years it was thought to be just that. Evolutionary leftovers. The accumulated debris of a billion years of life. The faded, meaningless footnotes of our own history. The library, it seemed, was 98% empty pages, 98% silence. The story of life, we thought, was contained only in those rare, illuminated chapters. But what if that assumption was wrong? What if the library wasn't mostly empty? What if we simply didn't know how to read the rest of the text? Could it be that in our focus on the beautiful, shining words, we had missed the deeper story being told in the quiet spaces? Could it be that the most important parts of a story are not the words themselves, but the silence that gives them shape and meaning? This vast, uncharted wilderness of our own DNA, dismissed as junk, was waiting. It held a secret, a paradigm shift in the making, and it is into this genetic darkland that we now must travel. Section 2. An unexpected wilderness. And so we stand at the threshold of a profound and unsettling revelation. The beautiful, orderly library we had imagined, with its neat rows of protein coding books, has dissolved into something far stranger, far wilder. Less a library and more an untamed wilderness. The discovery of this vast expanse of junk DNA was not just a small correction to the map of life. It was the realization that we had been reading the map upside down. Imagine the explorers of old, having crossed a great ocean, expecting to find a small, familiar island. Instead, they stumble upon a continent that stretches beyond the horizon, shrouded in mist, silent and inscrutable. This was the feeling in the world of genetics. The 2% of our DNA that coded for proteins, this was the familiar island. We knew its harbors, its pathways, its inhabitants. The other 98% was the new continent, a place for which we had no charts, no language, no guides. The term junk is a powerful one. It suggests something useless, broken, ready to be discarded. And for a time this was the prevailing view. This non-coding DNA was seen as the attic of the genome, a dusty, cluttered space filled with evolutionary relics. Here you might find the ghosts of ancient genes, sequences that were vital to a distant, water-breathing ancestor, now silenced and broken, like derelict ships on a sea floor. Here too were the viral footprints of millennia, the DNA of viruses that had injected themselves into our ancestors' genomes, becoming a permanent, though seemingly dormant, part of our lineage. Another idea that took hold was that of selfish DNA. This was a fascinating, almost subversive concept. Perhaps this extra DNA wasn't for our benefit at all. Perhaps it was a kind of genetic parasite. Simple, repetitive sequences whose only biological purpose was to replicate themselves, to make more and more copies, hitching a ride through the generations within our cells. They were like snippets of a song that gets stuck in your head, endlessly repeating, not because it's profound but simply because it can. From this perspective, our genome was not a perfectly honed machine, but a crowded ecosystem, home to squatters and hitchhikers who were just along for the ride. This notion that our own source code was mostly clutter and noise was deeply counterintuitive. Nature, especially at the molecular level, is typically a ruthless editor. It favors efficiency. It doesn't waste energy carrying around useless baggage. Yet here we were, with a genetic backpack seemingly stuffed with 98% rocks. To carry all this extra DNA, to copy it meticulously every single time a cell divides, from the first spark of life in the womb to your last breath, requires an enormous amount of energy. Why would nature, the ultimate minimalist, be so extravagant? It was a paradox that sat uncomfortably at the heart of biology. Let us return to our quiet, inner space. Feel the gentle rhythm of your own breath. With each inhale and exhale, trillions of your cells are humming with activity, and in many of them your DNA is being copied. Imagine the sheer effort, the molecular cost of replicating all of that junk over and over again. It is like a monk tasked with copying a sacred text, who is forced to copy not only the single meaningful sentence on each page, but all the supposed gibberish as well. Surely there must be a reason. Slowly, a new question began to be whispered in the quieter corners of science. The question was not, why do we have all this junk? But rather, what if it isn't junk at all? What if we are the ones who are mistaken? What if we are like a traveler in a foreign land who, unable to read the language, declares all the books and signs to be meaningless scribbles? The clues were subtle at first, like faint sounds in a dense forest. Scientists began to notice that some of these junk regions were surprisingly consistent across vast evolutionary distances. If you compared the genome of a human to that of a mouse or a fish, you would find that some of these non-coding sequences were almost identical. They had been preserved unchanged for hundreds of millions of years. Nature, you see, is a constant tinkerer. DNA sequences that are not important tend to drift and change over time, accumulating random mutations. But if a sequence is preserved, if it is locked in place across species that have been separated for eons, it is a powerful sign. It means that sequence is doing something important, something so critical that any change is harmful, and is weeded out by evolution. These conserved regions were like ancient, well-trodden paths through the wilderness. We didn't know where they led, but their very existence told us that someone or something had been walking them for a very long time. So the perspective began to shift slowly but profoundly. The junk was not a garbage heap. It was a landscape, and it had features. It had conserved pathways, it had strange repeating structures like mountain ranges, it had vast, quiet plains. It began to look less like an attic and more like the dark matter of the genome. In cosmology, dark matter is the invisible substance that makes up the vast majority of the universe. We cannot see it, but we know it's there because we can see its gravitational pull on the stars and galaxies we can see. Perhaps junk DNA was the same. Perhaps it was exerting a subtle, powerful influence on the shining, visible genes. Perhaps it was the hidden grammar, the punctuation, the syntax of the genetic language. A gene might be a powerful word like sun or ocean, but without grammar, a collection of words is just a list. It is the grammar that arranges them into poetry, into instructions, into a story. It tells you when to read a word, how loudly to say it, when to pause, and when to move on to the next. This was the new vision. The 98% was not silence. It was the operating system. The protein coding genes were like the applications on a computer, the word processor, the web browser, but the non-coding DNA was the vast, complex operating system running in the background, managing everything, allocating resources, telling the applications when to open and when to close. It was the hidden architecture that made the entire system work. With this new perspective, the wilderness no longer felt empty or meaningless. It felt full of potential. It was a place of deep mystery, a frontier of human knowledge. The age of simply listing the genes was over. The age of exploring the vast, dark territory between them had just begun. We were standing at the edge, peering into the mist, ready to map the genetic darklands. Section 3. Mapping the genetic darklands. So let us begin. We stand at the edge of this immense internal continent, this 98% of our own being that we have called junk. The mists of misunderstanding are beginning to clear, and with them, the dismissive name is fading. This is not a wasteland. It is terra incognita, unknown land. And like the explorers of any new world, our first task is to create a map. But how do you map a world you cannot see? How do you chart a landscape whose features are not mountains and rivers, but influence and information? We must become a new kind of cartographer, a cartographer of the invisible. Our tools are not the sextant and the compass, but powerful new ways of seeing and thinking about our DNA. Our parchment is not paper, but a new kind of understanding, a willingness to see purpose where we once saw only chaos. Imagine unrolling a vast, blank sheet. This is our new map of the genome. In the old cartography, we would have simply placed a few dozen thousand dots on itone for each gene and called the work finished. The vast emptiness between the dots would have remained blank, ignored. But now our task is to fill in that emptiness, to chart the very space between the genes. Our first act as cartographers of the Darklands is not to rush in, but to be still, to watch, to listen. We develop methods to see which parts of this vast landscape are active. We learn to look for footprints, for signs of life. We design molecular probes that can light up whenever a piece of this non-coding DNA is being read or used by the cell. And when we do this, a breathtaking picture begins to emerge. The Darklands are not dark at all. They are aglow with hidden activity. It is like looking at a satellite image of Earth at night. You see the familiar bright clusters of the great cities. These are the genes, always bustling, always shining. But now, for the first time, you see the intricate network of roads connecting them, the faint but persistent lights of small towns and isolated homes, the glowing shipping lanes across the dark oceans. The supposed emptiness is, in fact, threaded with light and purpose. Our blank map begins to fill. We are not just drawing dots anymore. We are drawing circuits, pathways, networks of influence. We see that many of these glowing regions are located right next to the known genes. They are like quiet neighbors, and we start to suspect they are not just neighbors but landlords. They seem to have some say over what happens in the bright house next door. So we draw a new kind of symbol on our map. Here, in this stretch of once ignored DNA, we sketch a symbol for a switch. Here, a symbol for a volume dial, a dimmer. These are the regions that will tell a gene when to turn on and when to turn off. They will tell a skin cell to be a skin cell by activating skin-related genes, while ensuring the genes for being a neuron or a liver cell remain silent and dark. They are the silent conductors of the genetic orchestra, and our map is now beginning to chart their positions. Then we must change our perspective. We must zoom out. For a long time we pictured DNA as a simple long string of letters, a piece of text to be read from left to right. But in the crowded, microscopic space of the cell's nucleus, this is not reality. The DNA is not a loose thread. It is a marvel of packaging and engineering. 2 meters of DNA must fit inside a space just a few millionths of a meter across. To achieve this, the DNA is folded, looped, and coiled in an incredibly complex and specific way. It is a form of genetic origami. Now we ask a new question. What if the folding instructions are written in the junk DNA itself? What if these vast non-coding regions are not just spacers, but the architectural struts, the hinges, the anchor points that shape the entire structure? We begin to map these features too. We draw loops and domains on our chart. We realize that a switch located millions of letters away from a gene on the linear string can, through this intricate folding, end up right next to it in three-dimensional space, touching it, whispering instructions to it. Our two-dimensional map is starting to leap into the third dimension. It is becoming a blueprint not just of a sequence but of a physical structure. And as we map, we also find curiosities. We find the ruins of an ancient past. Long repeating sequences that are, in fact, the fossilized remains of ancient viruses, invaders from millions of years ago, now silenced and woven into our own story. We find jumping genes, restless segments of DNA that have copied and pasted themselves throughout the genome over eons. For a long time, these were the poster children for junk or selfish DNA. But now, as we map their locations, we start to see a pattern. We find them often nestled inside the control regions, the switches and dimmers. And a radical thought occurs. What if these ancient invaders and restless wanderers are not just junk, but have been repurposed? What if the genome, like a resourceful artist, has taken this junk and used it as raw material for creating new controls, new complexities? We add another layer to our map, a historical layer, charting the ghosts and echoes of our deep evolutionary past. This map we are creating is the most intimate map of all. It is not the chart of a distant star or a forgotten corner of the Earth. It is the emerging map of the living landscape within you. It is a testament to the idea that there is no true silence in biology, only languages we have yet to decipher. The journey is no longer about finding the few shining islands of meaning. It is about understanding the ocean itself. Our compass is oriented. Our gaze is focused. We are ready to move from the abstract overview of the map to the tangible reality of the landscape. We are ready to approach the first of these glowing, mysterious features we have just charted, the silent conductors that stand guard over the genes. Section 4. The silent conductors. Let us now walk into the landscape we have mapped. We leave the high vantage point of the cartographer and descend into the intricate, glowing terrain of your own nucleus. We approach one of the bright islands of Mianinga gene, humming with potential, ready to build a vital protein. But our focus is not on the island itself. It is on the flickering lights we saw in the surrounding darkness, the regions of once called junk that lie nearby. It is here that we meet the silent conductors. Consider the great mystery of you. Every single cell in your body, with very few exceptions, holds the exact same library of DNA, the same 20,000 genes. The cell that makes up the lens of your eye has the blueprint for creating digestive enzymes. The cell in your liver holds the instructions for building the neurons that carry your thoughts. A skin cell, protecting you from the world, contains the genetic recipe for the hemoglobin that carries oxygen in your blood. And yet, they are not the same. A liver cell does not try to see. A brain cell does not try to filter blood. Each cell has committed to a unique identity. It reads only the chapters of the genetic book that are relevant to its task, while leaving all the others closed and silent. How does it know? Who tells the cell which page to read? For the longest time, this was a profound puzzle. The answer we have discovered is written in the dark lands. The decision is made not by the genes themselves, but by the non-coding DNA that surrounds them. These are the conductors of the cellular orchestra. They do not play an instrument themselves, they create no protein, but they control everything. They tap their baton, and a symphony of cellular life begins. Let us look closer at one of these regions. Imagine we are just upstream from a gene on the long thread-like molecule of DNA. This region is called a promoter. You can think of it as the ignition switch for the gene. The gene is the engine, but without a signal to this specific promoter sequence, the engine will remain cold and silent. The cell is flooded with countless roving proteins called transcription factors. Think of them as keys floating through the cellular space. Each one is a different shape, designed to fit a specific lock. The promoter region on the DNA is the lock. When the right key, the right transcription factor, finds its specific promoter lock, it clicks into place. This connection is the signal. Read this gene. Read it now. A complex machine then assembles at the site, and it begins to transcribe the gene into a message, the first step in building a protein. This is a simple on-off switch. But life is rarely so simple. It requires subtlety, nuance. You don't just want to turn a light on, you want to be able to dim it. You don't just want to start an engine, you want to be able to control the throttle. This is where the story gets even more wonderful. Let us now journey further out into the darklands, far away from the gene along the linear strand of DNA. We travel past thousands, even millions of letters of code that seem to have no function. And here, in this remote wilderness, we find another one of our glowing regions. These are called enhancers or silencers, and they are the true artists of gene control. These are the master volume dials. A promoter might be the on button, but an enhancer dictates how much, how strongly, and when that gene should be turned on. One enhancer might say, only turn on this gene when the cell is low on sugar. Another, controlling the same gene, might say, turn this gene on but only very weakly if a specific hormone is present. So now the cell's identity comes into focus. A liver cell will be filled with the specific keys, the transcription factors that fit the locks of the enhancers for liver genes. A neuron will be filled with the keys for neuron enhancers. The genes themselves are available in all cells, but the keys to activating them are different in each place. This is how specialization is born. But a new puzzle emerges. If this enhancer, this volume dial, is hundreds of thousands of letters away from the gene it controls, how does it send its signal? How can a conductor standing on the other side of a mountain command an orchestra? The answer lies in the genetic origami we spoke of, the beautiful folding of DNA. In the three-dimensional space of the nucleus, that vast distance vanishes. The DNA strand with its enhancer region loops around, folding back on itself in a precise and deliberate arc, bringing that distant enhancer right up next to the gene's promoter switch. Imagine taking a long ribbon, marking a spot at each end, and then tying a knot that brings those two spots together. This is what your cell does. And at that point of contact, a whisper of information is passed. The enhancer, now activated by its own key, touches the gene's starting machinery and says, begin. And begin with this much strength. Or in the case of a silencer, it touches the gene and says, be quiet now. It is not your time. Now picture this not for one gene, but for all 20,000. Picture not one or two enhancers, but hundreds of thousands of them, scattered throughout the vast non-coding genome. A single gene might be controlled by a dozen different enhancers, a committee of conductors that, together, create an exquisitely fine-tuned response. This is why your body can react so precisely to a changing world. It's why a wound heals, why your pupils adjust to light, why you can digest a meal. It is a constant, dynamic symphony of activation and silencing, orchestrated by these humble, powerful sequences in what we once called junk. We have found the grammar of the genetic book. The genes are the nouns and verbs, but the non-coding enhancers and promoters are the punctuation, the syntax, the staged directions that turn a simple list of words into a story of breathtaking complexity. They are the silent, invisible force that sculpts a single fertilized cell into the masterpiece of trillions of cells that is you. The Darklands are beginning to speak, and their language is one of control. Section 5, the Chromatin Architects. We have just witnessed an act of pure magic. A distant piece of DNA, an enhancer, reaching across a vast genetic space to touch a gene and command it to speak. That looping, that elegant arc that makes contact possible, is the secret to the cell's subtle control. But it begs a deeper question, one that takes us to the very physical nature of our being. What ensures the loop forms so perfectly? What prevents the DNA from becoming a hopeless, tangled mess? Who or what is the architect of this exquisite three-dimensional structure? Once again, the answer lies waiting for us in the vast, non-coding Darklands. To understand it, we must zoom in even closer, to see the DNA not just as a sequence of letters but as a physical object. Imagine again that 2-meter thread of DNA. It does not simply float freely inside the nucleus. That would be like stuffing 2 kilometers of fine thread into a tiny locket. It would not entangle instantly. Instead, the DNA is wound around millions of tiny proteins called histones. Picture the DNA as a shimmering thread, and the histones as microscopic spools. The thread wraps around one spool, then stretches a short distance, then wraps around the next and the next in a beautiful repeating pattern. This combination of DNA and its protein spools is called chromatin. It is the physical substance of your chromosomes. This winding is not just for storage. It is another profound layer of control. When the thread of DNA is wound tightly around its spools, the letters of its code are hidden, pressed against the protein. They are unreadable. The machinery of the cell cannot access them. A gene in a tightly wound region is a closed book. It is silenced, dormant. But when a gene needs to be read, the chromatin must change. The spools must loosen their grip. The thread must unwind, exposing its sequence of letters to the cellular machinery. An active gene is an open book, its story available to be read. So the control of life is not just about which keys, transcription factors, are present to turn on a gene. It is also about whether the book of the gene itself is physically open or closed. This brings us back to our architects. What determines which regions are open for business and which are locked down? Scattered throughout the non-coding wilderness are special sequences of DNA. These sequences don't control a gene directly, like an enhancer does. They have a different, more structural role. They are the tethering points, the boundary markers, the foundation stones of the genetic city. Let's focus on one type of these architectural sequences, known as insulators. Imagine them as small, powerful anchor points embedded in the DNA thread. Specific proteins in the cell recognize these insulator sequences and bind to them tightly, acting like powerful molecular clips. Now, imagine two of these insulator clips far apart on the linear DNA strand. The cell can bring these two clips together, forming the base of a loop. The stretch of DNA between them is now isolated, like a walled garden. Everything inside this loop, a gene, its enhancers, its promoters, can now interact with each other. But they are insulated from the affairs of the neighboring loop. These defined loops are called topologically associating domains, or TADs. They are the fundamental neighborhoods of the genome. It is these neighborhoods that solve the puzzle of the looping enhancer. The enhancer and the gene it controls are built into the same loop, the same walled garden. When the time is right, the DNA inside this domain can move and flex, allowing the enhancer to find its target. But it is prevented by the insulator boundaries from accidentally reaching out and activating a gene in the next neighborhood over. The junk DNA, therefore, provides the blueprint for the physical city of the genome. It says, "This is the financial district where genes for metabolism will be active. This is the arts district, where genes for creativity and complex thought reside. And here we will place a wall." These insulator sequences, these simple lines of code in the non-coding darklands, are the architects that draw the city map. And this architecture is alive. It is not a static blueprint of concrete and steel, but a fluid, dynamic structure. As the cell responds to new signals, as it grows and changes, this chromatin architecture can reshape itself. New loops can form, old ones can dissolve. Regions that were once tightly packed and silent can unfurl and open, while others are shut down and put into deep storage. It is a living, breathing origami, constantly refolding to meet the demands of the moment. So we have peeled back another layer of the mystery. The 98% is not just home to the conductors who direct the orchestra. It is also home to the architects who designed the concert hall itself. They have built the stage, arranged the seating, and soundproofed the walls, ensuring that every note is heard clearly, in the right place at the right time. The physical shape of your DNA is a form of information, as critical as the genetic code itself. And this information, this architectural genius, is written in the language of the genetic darklands. Section 6. Ghosts in the Machine. We have explored the living, breathing architecture of the genome, the loops and domains that form the city of the cell. We have met the silent conductors who direct its daily life. But now we must become not architects but archaeologists. For this landscape we are exploring is not new. It is ancient. It is built upon the ruins of forgotten worlds, and it is haunted, in the most beautiful sense of the word, by the ghosts of a time long past. To truly understand the Darklands, we must dig. We must uncover the stories buried deep within our own code. When we do this, when we analyze the sequences in these vast non-coding regions, it is a gene that came from an ancient retrovirus. The virus used this gene to fuse cells together to help it spread. Our ancestors co-opted this viral gene, this ghost in the machine, and used it to create one of the most essential innovations in human biology. Without this ancient viral infection, our form of life might not exist. Now let us turn to another kind of ghost, not an invader from the outside, but a wanderer from within. Throughout the genetic darklands, we find stretches of DNA that seem to have a life of their own. They are called transposons or jumping genes. These are restless sequences that have the ability to copy themselves and insert that copy into a new location in the genome. For decades, these were viewed as the ultimate genetic parasites, selfish bits of DNA whose only drive was their own proliferation, cluttering up the genome with copies of themselves. They were seen as a source of chaos, occasionally jumping into the middle of a vital gene and breaking it. But the story of the transposons is also undergoing a profound revision. We are finding that these wandering ghosts have been a powerful engine of creation and evolution. When a transposon jumps into a new location, it can bring with it a piece of regulatory code like a small enhancer switch. Suddenly, the gene near this new landing site has a new conductor, a new instruction. This can dramatically alter how the gene functions, creating new traits for evolution to act upon. These jumping genes have shuffled the deck of our genetics for millions of years. They have created new control circuits, new ways of wiring our genes together. They are not just chaos. They are a source of novelty, of innovation. They are the wild cards of the genome. The restless spirits whose wandering has helped to sculpt the complex creature that you are. So, as we drift through the inner landscape of your cells, we see that it is not a clean, sterile workshop. It is haunted. It is inhabited by the spirits of ancient viruses and the echoes of restless, jumping genes. But these are not malevolent spirits. They are ancestral ghosts who have become part of us. They are woven into our regulation, our development, our very identity. They remind us that the genome is not a perfect design machine. It is a product of history, of accident, of invasion and co-option. It is a beautiful, messy, living collage, and its story is written not just in the bright ink of the genes, but in the faded, ghostly script of the Darklands. Section 7. The Shadowy World of RNA. For our entire journey so far, we have been focused on the DNA itself, the grand ancient library, the master text. We have seen how its non-coding regions act as architects and conductors, building the physical structure and directing the symphony of the genes. But a library, no matter how well organized, is a silent place. A blueprint, no matter how detailed, cannot build a house on its own. Information needs an agent. It needs a messenger, an actor, a force to carry its intentions into the living world of the cell. You might remember a simpler story, the one we all learned. In this story, DNA is the king, safe in his castle of the nucleus. To get a message to the workers in the cytoplasm, the king transcribes his orders onto a disposable scroll. This scroll is called messenger RNA or mRNA. It is a temporary copy of a single gene, a single blueprint, sent out to the cellular factories. The ribosome must be read and translated into a single protein. In this old story, RNA was just that, a simple courier, a fleeting message with no life of its own. But as we began to explore the genetic darklands, a parallel and equally profound mystery was unfolding. When scientists developed ways to see all the RNA being made in a cell, they expected to find mostly mRNA, the scrolls copied from the 20,000 known genes. Instead, they found a reality that was vastly more complex and beautiful. They found that the cell was transcribing enormous portions of the junk DNA. The Darklands were not silent at all. They were being read, constantly, passionately, and turned into countless RNA molecules that were not messengers destined to become proteins. Welcome to the shadowy world of non-coding RNA. This discovery was like realizing that in a bustling city, the official messengers and couriers make up only a tiny fraction of the traffic. The streets are actually filled with secret agents, guides, saboteurs, and managers, all communicating in a code we never knew existed. These non-coding RNA molecules are the active agents of the Darklands. They are not the blueprint. They are the workforce that interprets the blueprint. Let us meet some of these shadowy operatives. First, there are the microRNAs or miRNAs. As their name suggests, they are tiny. Imagine a short, elegant snippet of RNA, just a couple dozen letters long. These are not messengers, they are molecular assassins. Once a gene has been transcribed into an mRNA scroll and sent out into the cell, the story is not over. An miRNA, itself transcribed from a region of junk DNA, can be waiting. It patrols the cytoplasm, and if it finds an mRNA molecule with a matching sequence, it binds to it. This binding is a death sentence for the message. The miRNA can either block the mRNA from being read by the protein factories, or it can mark it for immediate destruction. This is a layer of control of exquisite subtlety. It's like having a final editor who can, at the last second, cross out a sentence or an entire paragraph before it's published. The cell can turn a gene on, but then use these tiny RNA operatives to fine-tune exactly how much protein is made from its message, or to shut it down quickly if conditions change. They are the guardians of cellular quiet, ensuring that no message shouts too loudly. Then there are the long non-coding RNAs or LNCRNAs. These are much larger, more complex molecules, and their roles are only just beginning to be understood. They are the true master organizers, the versatile project managers of the genome. They are the secret agents who can walk between worlds. Imagine a long strand of RNA, transcribed from a deep and forgotten corner of the Darklands. It folds itself into a unique and complex three-dimensional shape. This shape is its function. This LNCRNA can act as a molecular scaffold. It might have one binding site that grabs onto a specific protein, one of our keys, or transcription factors, and another site that recognizes a specific address on the DNA. The LNCRNA then acts as a guide, a shepherd, physically carrying that protein through the crowded nucleus and delivering it to the exact spot on the genome where it needs to act. It is the GPS of the cell. Other LNCRNAs act as decoys. The cell might want to silence a whole group of genes, so it transcribes a LNCRNA from the Darklands that is covered in binding sites for a specific on-switch protein. This LNCRNA floats through the nucleus, and all the target proteins, instead of finding the genes they are meant to activate, bind to the LNCRNA decoy instead. They are captured, neutralized, their power soaked up by this shadowy molecule. And perhaps most profoundly, these LNCRNAs are intimately involved with the chromatin architecture we spoke of. They can bind to the machinery that opens and closes the DNA, guiding the architects, telling them which neighborhoods of the genetic city to place on lockdown and which to open for business. So you see, the genetic Darklands are not just a static set of switches and anchors. They are the source code for a dynamic living network of RNA molecules that pervade every corner of the cell's life. The DNA may hold the plan, but it is this shadow world of RNA that executes it. They are the software running on the hardware of the chromatin. They are the whispers, the guidance, the subtle acts of sabotage and management that translate a static library of genes into the dynamic, responsive, living thing that is you. The darkness is alive with their voices. Section 8. The Hidden Operating System. We have journeyed far. We began in a library that seemed mostly empty, then found ourselves in a vast wilderness. We learned to map this new continent, discovering its features were not random, but were the work of silent conductors and masterful architects. We dug into its soil and found the ghosts of an evolutionary past. And we have just witnessed how this silent landscape gives rise to a shadowy world of active RNA agents, executing a will we never knew existed. Now is the moment to step back, to ascend once more to a high vantage point and see how all these pieces, the switches, the loops, the ghosts, the RNAs fit together. For they are not separate stories. They are chapters in one single magnificent narrative. They are the components of a system so complex and elegant that we are only just beginning to appreciate its genius. It is time to name this system. The genetic darklands, the 98% of our DNA we once dismissed as junk, is the hidden operating system of life. This is the central revelation of our journey tonight. The old idea of the genome as a simple list of instructions, a book of recipes, is profoundly incomplete. It is like looking at a powerful computer and seeing only the icons on the screen, the applications. You see the icon for build a heart cell, the icon for digest sugar, the icon for think a thought. These icons are the protein coding genes. They are important, of course. They are the programs that do the work. But what launches the program? What tells it when to run, how much memory to use, how to interact with other programs? What happens when you, the user, click the icon? In the background, unseen by you, the computer's operating system springs into action. It is a vast, intricate scaffold of code that manages the hardware, allocates resources, and makes it possible for the applications to run at all. You never see the operating system, but without it, the icons on the screen would be just static, useless pictures. This is the role of the 98%. It is the deep foundational code. Let us imagine booting up the computer of you. First, the most basic low-level instructions are loaded. This is the BIOS, the firmware of life. These are the architectural sequences in your non-coding DNA insulators that define the very structure of the hard drive, organizing the data into fundamental loops and domains, the TADs. These are the centromeres that manage the physical duplication of the chromosomes. This code is ancient, stable, and absolutely essential. It lays down the physical hardware configuration before anything else can happen. Next, the kernel and the system drivers begin to load. These are the background processes that manage the entire system. In our genetic OS, this is the world of the long non-coding RNAs and the chromatin remodeling proteins they guide. They are constantly running, assessing the state of the system. They are the memory managers, physically opening and closing regions of the DNA hard drive, deciding which applications, genes, are accessible and which are archived in deep storage. They are the communication drivers, ensuring that a signal from the outside world, a hormone, a nutrient, a ray of light is correctly routed to the appropriate software. Now, with the system running, the user interface appears. These are the enhancers, the promoters, the silencers. They are the buttons, the dials, the menus of the genetic operating system. They provide a simple, elegant way for the cell to perform incredibly complex tasks. A specific transcription fact or a key floating in the cell arrives. This is the user making a choice. It binds to an enhancer, it clicks a button. When that happens, the user doesn't need to know the details. The operating system takes over. It verifies that the chromatin in that region is open. It ensures the correct looping architecture is in place to connect the enhancer to its target gene. It deploys the RNA polymerase machinery to begin transcription. A whole cascade of complex background events is initiated by that one simple click. The application begins to run. And what about the ghosts in the machine? The ancient viruses and jumping genes? In our OS analogy, they are like legacy code, or perhaps even bits of code copied from another programmer's open-source project. Over millions of years, the operating system has repurposed this old code. It found a useful subroutine in a piece of ancient viral software, and patched it into its own system to run a new function, like building a placenta. It used the random insertions of jumping genes as a way to beta test new connections in its user interface, creating new buttons and links where none existed before. This explains the great paradox we started with: why a human with a mere 20,000 genes can be so vastly more complex than a rice plant with 50,000. It is not about the number of applications. It is about the sophistication of the operating system. Our 98% is filled with layers upon layers of regulatory control, a richer user interface, more complex background processing, a more dynamic and responsive system architecture. We don't have more programs. but our OS can run them in infinitely more subtle and powerful combinations. It allows for the creation of a brain of consciousness of art not by adding new genes, but by adding new depths of regulation to the genes we already have. The genome is not a book. It is a living, self-regulating, computational device of unimaginable power. And its genius lies not in the shining applications we have always seen, but in the vast, dark, silent operating system that has been running in the background all along. Section 9. When the system glitches. For our entire journey we have marveled at the elegance of this hidden operating system, the flawless choreography of the conductors, architects, and RNA agents. We have seen a system of breathtaking complexity that works in silent perfection. But as with any operating system, whether in a computer or a cell, glitches can happen. Bugs can appear in the code. And in the world of biology, a bug in the operating system is not a minor inconvenience. It doesn't just cause the screen to freeze. It can lead to disease. This is the newest and perhaps most clinically important chapter in the story of the Darklands. For decades, when doctors and scientists searched for the genetic roots of disease, they looked almost exclusively in one place: the 1 to 2% of DNA that contained the protein-coding genes. They searched for spelling mistakes or mutations within the blueprints. And they found many. Diseases like cystic fibrosis and sickle cell anemia are caused by clear, definable errors in a single gene. The recipe for a vital protein is wrong, and the body suffers. But for many of the most common and complex diseases that afflict humanity, conditions like heart disease, diabetes, autoimmune disorders like lupus and Crohn's disease, and many forms of cancer, the search for a simple single-gene cause proved frustratingly elusive. There were hints of a genetic link, as these conditions often run in families, but there was no single broken gene to point to. The blueprints in many cases appeared to be perfect. The mystery was immense. How could a genetic disease exist without a broken gene? The answer we now know was hiding in plain sight in the 98% we had ignored. The problem wasn't a flaw in the application software. It was a glitch in the operating system. Let us return to the quiet landscape of your nucleus and imagine what happens when a single letter is misspelled. Not in a gene, but in the non-coding DNA. Imagine a mutation, a tiny error, occurring in the sequence of an enhancerone of our volume dials. The enhancer sequence is now slightly altered. The key, the transcription factor that is supposed to bind to it, no longer fits perfectly. The result can be subtle but catastrophic. Perhaps the key can still bind, but not as tightly. The volume dial is now loose. A gene that should be turned on to a precise level of 5 is now only turned on to a level of 2. Or maybe the opposite happens. The mutation makes the enhancer more powerful. A gene that should be whispered is now shouted. The delicate balance is broken. Think of a gene that helps to control cell growth. In a healthy person, the enhancers for this gene tell it to be active only when the body needs to create new cells, for instance to heal a wound. When the healing is done, the enhancers are silenced, and the gene becomes quiet again. Now, imagine a mutation in one of these enhancer sequences. A glitch. Suddenly, the off switch is broken. The enhancer is stuck in the on position. The gene that promotes cell growth is now active all the time, whispering grow, grow, grow without end. This is one of the ways that cancer can begin. Not with a broken gene, but with a broken control switch in the Darklands. Or consider an autoimmune disease. Your immune system is a marvel of precise control. It must be able to recognize and attack foreign invaders like bacteria, while leaving your own healthy cells untouched. This identity check is managed by an incredibly complex network of genetic switches. Now, imagine a mutation in a silencer region that is supposed to keep a powerful attack gene silent in the presence of your own tissues. If that silencer breaks, the gene might turn on at the wrong time in the wrong place. The immune system, reading this faulty instruction from the operating system, can fail to recognize itself. It begins to attack your own body. These glitches can also happen in the architectural code. A single spelling mistake in an insulator sequence one of our molecular clips can weaken it. The anchor point fails. The carefully constructed loop, the walled garden, falls apart. Suddenly, an enhancer that was meant to control a gene inside its own neighborhood can now reach out and touch a gene in the next domain over, one it was never meant to interact with. It's like a short circuit in the computer's motherboard, causing unpredictable and chaotic behavior. This new understanding is revolutionizing medicine. Scientists are now hunting for the genetic roots of disease not just in the genes, but in the vast, non-coding genome. They are comparing the darkland DNA of thousands of individuals, looking for these subtle single-letter variations that are more common in people with a particular disease. These are called genome-wide association studies, or GWAS, and they are providing a completely new map of human disease, a map that charts the glitches in the operating system. It is a more complex view of illness. It tells us that for many conditions, there isn't a single cause, but an accumulation of tiny, subtle risks, written in the quiet language of our genetic controls. But with this complexity comes a new kind of hope. If we can understand the glitches in the operating system, we may one day learn how to fix them, how to rewrite the faulty code, and restore the beautiful, delicate balance that is the hallmark of health. Section 10. An invitation to wonder. And so, we arrive at the edge of our map, at the end of this particular path through the great inner wilderness. We have traveled from a place of seemingly simple certainty to one of profound and beautiful mystery. We began our journey in a library we thought we understood. A library with a few precious books called genes, surrounded by what we believed was meaningless silence. We end our journey in a world humming with the language we are only just beginning to decipher. The story of junk DNA is not just a story about science. It is a story about how we see the world and how we learn to see it anew. It is a parable of humility. For decades, we looked at 98% of ourselves and called it junk, a testament to our own limited imagination. We projected our own ignorance onto the elegant text of life and declared the text to be at fault. We were like a reader who, finding a book written in a language they do not know, declares the book to be filled with nonsense. But now the book is beginning to open to us. The junk has revealed itself to be the very soul of the system. It is the conductor's score, the architect's blueprint, the historian's record, the manager's software. It is the source of the subtlety and grace that allows a single genetic code to build a being capable of looking back upon that code with wonder. What we have discovered is that the genome is not a simple list. It is a poem. The genes are the words, but the non-coding DNA is the meter, the rhyme, the structure, the metaphor. It is the silence between the notes that creates the music. And it is this vast regulatory landscape that creates the symphony of you. This journey invites us to hold a new kind of wonder for ourselves. As you lie here, breathing in the quiet of the room, sense the universe of activity within. In trillions of cells at this very moment, this hidden operating system is running. DNA is looping and unlooping in a dance of architectural precision. Long non-coding RNAs are acting as shepherds, guiding proteins to their homes. Enhancers are listening for the subtle chemical signals of your body, adjusting the expression of your genes in real time. The ghosts of ancient viruses are lending their strength to the daily functions of your life. This is not a chaotic, random process. It is a performance of immense, and for the most part, silent perfection. The sheer depth of this complexity is a powerful meditation. It tells us that life is not a simple, deterministic machine, easily reverse-engineered. It is a layered, historical, evolving masterpiece. It built itself over billions of years, repurposing junk, domesticating invaders, and writing new layers of code upon old. You are the current living expression of that 4-billion-year-old story. And perhaps the most beautiful realization of all is that this story is nowhere near its end. For every question we have answered about the genetic darklands, 1,000 new ones have blossomed. We have mapped the coastlines, but the deep interior of the continent remains largely unexplored. We know there are long non-coding RNAs, but we have only identified the function of a tiny fraction of them. We know the genome folds into loops, but we do not yet fully understand the grammar that dictates this folding. We have found the glitches that lead to disease, but we are only at the very beginning of learning how to gently debug this living code. We have not solved the mystery. We have only discovered its true scale. And that, in itself, is the greatest discovery of all. It is an invitation to remain curious. It is a promise that there will always be more to learn, more to explore, more to be amazed by. The book of life is far grander and more mysterious than we ever dreamed, and the best chapters may be the ones we have not yet learned how to read. We are standing at the dawn of a new understanding, peering into a world of exquisite complexity that is not in a distant star or another dimension, but is folded carefully and precisely within each and every one of us. And so our journey through the genetic darklands comes to a gentle close. We leave the conductors and architects to their silent, ceaseless work. The story we have uncovered tonight is not one to be memorized, but to be felt, a sense of the deep, quiet, and profound intelligence that animates the very fabric of your being. It is the ultimate lullaby, the hum of an ancient operating system that has been running perfectly since the moment you began. Let this feeling of intricate wonder be a soft pillow for your mind. There is nothing more to do, nothing left to explore for now. There is only the peaceful, rhythmic certainty of the life within you. May your sleep be as deep and peaceful as this hidden world. May your dreams be filled with quiet wonder, and may you awaken refreshed, carrying a small sense of the beautiful, complex miracle that you are. Good night.